Thank you, Georgia, for that beautiful talk, and for the organizers for inviting us and letting us present our work. We're so excited to share this with you. Oh, I have the spinner of doom, sorry. Okay. My name is Elina Kostyanovskaya, and I'm a graduate student in Dr. Helen Wilsey's lab studying autism genetics using frogs as a model organism. And today I'll be talking about the role of SYNGAP1 outside of the synapse at the cilium. So recently, thanks to rare variant-based DNA sequencing, a large number of high-confidence, large-effect autism genes have been identified. And a variant in any one of these top genes increases the likelihood of autism several-fold. SYNGAP1 is one of these high-confidence, large-effect autism genes, and while it has been known that autism commonly co-occurs with cilia-related conditions such as congenital heart disease, hydrocephalus, blindness, epilepsy, and more, the possibility that autism proteins function at the cilium has not been explored. And so based on these observations and some previous work in the lab, we hypothesized that ciliary biology is relevant to autism biology. And so a little bit background on cilia. Cilia are these hair-like structures found on almost every cell, including neurons, and a single primary cilium acts like the cell's antenna. It's located right near the soma or nucleus, and it consists of this basal body from which the cilium nucleates, and then the axoneme and microtubule-based projection. And so the primary cilium is important for signaling, especially during development. It integrates extracellular signals in neurons and regulates the balance of excitatory and inhibitory signals in the brain. And many proteins and receptors are localized specifically to the cilium, and different proteins are localized to the cilium depending on the cell type, i.e. neurons, and cell state. And so changing the localization of proteins specifically at the cilium affects processes such as development and neuronal signaling. So, for example, decreasing the localization of somatostatin receptor 3 to the cilium actually alters the excitatory to inhibitory balance in the brain. MC4R localization is also associated with obesity and altered feeding, and dopamine receptor 1 actually localizes to the cilium, and work from about 2 years ago shows that SYNGAP1 is known to interact with dopamine receptor 1. So cilia actually come in two main flavors. It's this primary cilium that I just talked about, which is for signaling and sensing, like in neurons. And it's also important in development and neurogenesis, and as well as multiple motile cilia that beat, like in lungs, and those are important for fluid flow and heart development. So to determine whether SYNGAP1 and other autism proteins have a role at the cilium, we performed a localization screen in which fluorescently tagged human autism proteins, including SYNGAP1, were expressed in our model of primary cilia, which are these rat striatal neurons, and as well as in Xenopus for motile cilia on the epithelium of Xenopus embryos. And as a result of the screen, we localized SYNGAP1, both endogenous and exogenous SYNGAP1, to the primary cilia of rat striatal neurons that we got from Dr. Ehrlich, and to the motile cilia of the frog epithelium. So just to walk you through this a bit, because I'll be showing these slides sort of throughout, the cilia here is labeled in magenta, and then you have the autism protein in green. And so here you're looking at endogenous SYNGAP1 in rat striatal neurons. And then when we express human SYNGAP1 in these rat striatal neurons, again, you see here the autism protein in green along the axoneme, and here is actually the basal body, and the cilium in magenta. And same thing with the motile cilia. Again, we have endogenous SYNGAP1 in these multiciliated cells, which are, again, the axonemes are labeled in magenta, autism protein in green, and then again, the human SYNGAP1 in multiciliated cells, again, here at the base and along the axoneme, and then you have the cilia here in magenta. So, oops. So I also localized endogenous SYNGAP1 to the primary cilia of human retinal pigment epithelial cells and to iPC-derived cortical neurons, as well as motile cilia endogenously in the ependymal cells of the rat brain. So these are the cells that beat and are thought to move cerebrospinal fluid. So you have SYNGAP1 expressed in primary cilia and motile cilia across cell types and across species. And in terms of humans, we actually find that it's almost as highly expressed in multiciliated cells as it is in neurons. So here you have all the different neuronal subtypes in a human transcriptomic data set in red, and then you have multiciliated cells in blue, and you can see that expression of SYNGAP1 in multiciliated cells is actually quite high in humans. And furthermore, we looked at recently published single-cell sequencing data from the developing mouse airway and find that SYNGAP1 is actually specific to the multiciliated cell lineage. And so the multiciliated cell lineage is actually denoted by FOXJ1 expression. So SYNGAP1 is on the cilium and it's highly expressed in human ciliated cells, but is it actually functional there? So to get at this, I knocked down SYNGAP1 two ways, one with CRISPR-Cas9 and one with morpholino, and you observed cilia defects. So the way that you can see that here is you have these sort of long cilia in magenta, but in a SYNGAP1 knockdown, either by CRISPR or morpholino, you can see this sort of loss of cilia and the uneven spacing. And so these cilia defects that we actually quantified can be rescued by reintroducing human SYNGAP1 into these knockdown Xenophus models. One other thing I'd like to point out here is actually you can see here in white, that white labeling is the phalloidin, which represents the actin network. And you can see that it's pretty substantially disrupted, which was validating to see because this is consistent with previous work showing that SYNGAP1 regulates actin dynamics. So next we wanted to see if autism-associated missense variants affect cellular localization of SYNGAP1. And so for these experiments, we use a SYNGAP1 alpha 2 because it's the only C-terminal isoform that contains all reported autism patient variants. And this isoform, unlike alpha 1, lacks the C-terminal PDZ binding domain, but it retains the coil-coil domain, which will be relevant in a second. So just to orient you, the coil-coil domain is here, and normally you'd have the PDZ binding domain here at the end. And so again, when we look at this wild-type SYNGAP1, we see that it's at the base and along the axoneme. However, when we look at this first variant, we see this pretty substantial mislocalization away from the cilium to the plasma membrane. And when we look at the second patient variant, we find that it also mislocalizes away from the cilium into sort of these liquid-like aggregates. And this was especially interesting given that these mutations we started with are within or adjacent to the annotated plextron homology domain, this little domain here, which is known to have a role in membrane localization. And so SYNGAP1 protein is thought to trimerize through this coil-coil domain. So it's very possible that these variants of SYNGAP1 are bringing or sequestering the wild-type SYNGAP1 to a different location. So for example, if you have 3 copies of SYNGAP1 and 2 of those are variant, the question is, can the 2 variant copies bring the wild-type SYNGAP1 to the wrong location? And so we can actually test this in frogs, and to do this, we take the GFP tagged reference SYNGAP1 and we co-express it either with another copy of the reference SYNGAP1 or with a variant. And so we find that here, when I express the reference SYNGAP1 with reference SYNGAP1, we get localization that's pretty normal to the base and the axioms of cilium. However, when you express them with that first variant, you start to see mislocalization of the reference SYNGAP1 away from the cilium to the plasma membrane. And again, when you co-express with the second one, you see mislocalization away from the cilium into these sort of liquid-like aggregates. So far, altogether, this work suggests that SYNGAP1 has a required function on the cilium, but does that translate to cilia defects in patients with de novo heterozygous variants? And so we looked at cohorts to see whether they had evidence of ciliary dysfunction. And through Citizen, we got access to a comprehensive data set of patients with de novo pathogenic variants in SYNGAP1. And when we look at this patient data, indeed, we see patients have skeletal defects, dysmorphic features, and structural heart defects. And looking at these heart defects in particular, of the heart defects, many of these are congenital structural heart defects or heart murmurs, which would be difficult to explain by a synapse-only mechanism, that is, if SYNGAP was only functioning at the synapse. But this is medical record data. If patients actually have a ciliary problem, we should be able to see that in real time. And so just a little bit of background in your nose, you have these nasal multiciliated cells with cilia that beat. And in the cases where there's cilia defects, this beating is actually important for producing nasal nitric oxide gas. And so when you have cilia defects, you actually have less nasal nitric oxide, and you can measure that in patients with primary cilia dyskinesia. So we went out, we bought a nasal nitric oxide machine, which is a 30-second non-invasive test, and we had James administer it. And we actually mostly largely at this conference last year. And we found that nasal nitric oxide levels of 24 patients with de novo variants in SYNGAP1 had significantly lower levels of nasal nitric oxide compared to unaffected family members. And this is consistent with SYNGAP1 having a ciliary contribution. And I also want to flag, James is actually here this year. James, can you wave your hand? Thank you so much. We're doing more nasal nitric oxide measurements. So one, we're really interested in getting more unaffected family members, but we would also like to get nasal nitric oxide measurements from people who got them measured last year. We want to see how nasal nitric oxide changes over time. So even if you got measured last year, please come by again and let us measure you again. Okay. So I also want to plug another senior graduate student that Georgia also mentioned in the lab, Kate McCluskey, who had done beautiful work at the intersection of SYNGAP1 and the gut, and showed that the loss of SYNGAP1 results in developmental issues, including gut neuron migration defects. And she also develops a new assay for gut motility, looking at the effects of SYNGAP1 loss on gut motility. She has a poster, so it's really cool work, so please check it out. And also just the migration of these neurons is known to be dependent on cilia. So this is another context in which ciliary biology might be important. And so just to wrap up this talk, that we know that SYNGAP1 is expressed in ciliated cell types. It localizes to and it functions at the cilium, despite annotations or known function at the synapses. We know that SYNGAP1 patient variants mislocalize the protein away from the cilium, that individuals with SYNGAP1 variants have conditions that are consistent with a ciliary contribution, and then individuals with SYNGAP1 variants have reduced nasal nitric oxide compared to family. And so, as I kind of alluded to at the beginning of the talk, SYNGAP1 is one of many autism proteins that are annotated as proteins involved in neuronal communication. And there's two main sort of functional annotations for autism proteins. One is in neuronal communication and one is in gene expression regulators. And we don't think that this role of autism proteins at the cilium is specific to SYNGAP1. We think it's actually a bunch of different autism proteins, and that is sort of where autism genetics converges. And in fact, we do see that, that if you look at all the other autism proteins, many of them localize to and function at cilium. And so this finding extends beyond SYNGAP into autism proteins more broadly. So to conclude, I want to leave you with the idea that, of course, proteins have a canonical role at the synapse, but there's also an opportunity here to think more deeply about the similarities between the synapse and the cilium. So both the synapse and the cilium have extravesical trafficking. They are rich in GPCRs and cytoskeletal components. They have a diffusion barrier at the base, and many proteins are actively transported through this along microtubules. They both regulate neuronal excitability, and they both modulate downstream pathways. And these similarities were first flagged by the Syngap lab about 10 years ago. And so with that, I want to thank the Syngap families who participated in and support this research, to the organizers for putting together such a wonderful conference, and the Syngap Research Fund for funding our work. A big thank you, obviously, to the Wilsey Lab, especially Helen for all her support, and James for the nasal nitric oxide work. I also want to thank the State Lab, the Writer Lab, and Dr. Ehrlich for their work on rat startle neurons. And also thank you to the funders, especially the NIH F99K00 mechanism that funds my work. So thank you very much.